Let me provide you just really few words about the mission of our research in our lab that is focused on data management with the final aim to find within very large data collection those objects that are relevant for the user and the services at hand. And in doing this, we focus on both effectiveness and efficiency aspects. We had a long experience with different types of data, from traditional data to non-conventional data, like, say, text documents, data stream, time series, images, video, and any kind of interesting pattern. And today we focus on data, genomic data and genomic pattern. I will show you how we were able to exploit our long expertise in terms of query processing algorithm based on the notion of similarity search within the Gen Data 2020 PRIM project, with a strong and tight collaboration with Politecnico of Milano for the bioinformatics and biological support. So, really quickly, my outline. After some motivation of the problem, I will introduce the problem we tackled that we call the best match problem, together with a base solution that consists in an optimized pattern matching algorithm. And I will present so many different extensions of the base solution, together with an experimental evaluation. And finally, I will show you how our algorithm, on top of our algorithm, we develop a desktop application that we call SimSearch as an IGB plugin. So, all of us well know what DNA sequences is and how the knowledge of DNA sequences has become so important for basic biological research in so many different applied fields, such as medical diagnosis, biotechnology, and so on and so forth. And so far, as stressed by Stefano in the previous talk, bioinformatics has been challenged by next generation sequences, primary analysis with the final hand to produce sequences in the form of reads or tracks, and secondary analysis with the goal to align such reads and reach with feature. But now the most important problem nowadays from our point of view is the tertiary analysis in terms of multi-sample processing, annotation of variants, genome browser-driven exploratory analysis, and we are focused on tertiary analysis. We tackle the data produced by the secondary analysis, so we are working with aligned data enriched by features, and with the final goal to extract the value from such data, so trying to provide a good answer to the big questions, so how genomic regions interact with each other. So, first of all, what a pattern is and why is it important searching for pattern in the genomic sequence. So a pattern can be defined as a co-occurrences of a certain region configuration. From a biological point of view, we can think about a pattern like a set of regions, peaks, at a given distance from each other along several tracks of the whole genome sequence. So, and why once the biologists have visually identify a pattern of interest in the whole genome, is important to find for similar occurrences in the whole sequence. It's important because it can help in understanding the biological meaning of NGS experiment, and even in understanding biomolecular phenomena. So it's so important for the tertiary mission. We can define our problem in this way. So as I show you in the figure, given a pattern, in this case, in the red part of the figure, we have a pattern that is formed by 6 regions spread in 3 tracks, and the genomic sequence, 5 regions in the sequence which are arranged as in the pattern, so find similar pattern. So this is a so important problem from the biological point of view, but it is even such complex computational task not currently supported. So we try to tackle this such complex problem that belongs to the class of quadratic assignment problem in order to shape it in a tractable way, as I will show you. So we start with the base problem where patterns are defined in single track, region are modeled as points that represent the coordinate, the linear genomic coordinate. So we work with pattern and track as sequences of integers. So here in red, again, we have an example of simple track pattern, and we have an example of target track matching, for example. What is important here is that we need what is important is the relative distances between elements of a pattern, just because we want to model the structure of our pattern. So we are talking about occurrences, co-occurrences. We formally define the occurrences in terms of matching. And matching is a strictly increasing function that assigns to each pattern element a track element, preserving the element order, because we are working with alignment regions. So this is an example of good match, and this is an example of bad match because it doesn't preserve the order. So with this in mind, we are ready to present our base solution that is based on a cost-based model, cost-based approach that is a quite usual approach in similarity search. So what we want to do is to define a cost as the sum of distances relative to the first matching pair, let's say the first one. So everything is computed with respect to the first match of our pattern. What is important in the formula of the cost is the inner element that represent the root matching and that we call root distance, that allows us to consider shifted offset in our cost model. And what is another key point of our cost model that allows us to get tractable such complex problem is that we were able to disjoin the computation of the root matching with respect to the computation to other matchings. So with the final goal here is to find the matches that minimize the cost, the overall cost, by minimizing, of course, in the matching, the difference, the distance between elements of the pattern and the region of the track. So let me provide an example. Here we have an example of pattern, query pattern, and the track. This is a possible matching for each region of the query with in the track, with root distance 2 and the overall cost 21, but this is not the best matching because the best matching is the one that minimizes the cost. That is the second one with the cost of 0 and root distance 4. So based on this model, on this cost model, we developed what we call the windowed dynamic programming algorithm that is able to solve the problem in logarithmic time complexity, which is the main result. First of all, we found a really nice theoretical main result that says that a matching is optimal if and only if all its partial matchings has minimum cost. And so based on this theoretical result, we can say the basic idea of the algorithm is that for all possible root positions, so for all possible root matching, and for all region of the pattern, what we do is to find, first of all, the best matching for each element of the pattern, using binary search, of course, and in case of conflict, so which means that in case we have a region of the genome target sequence that is matching with multiple regions in the pattern, we enlarge the windows in order to solve in an increasing way the problem. So the key idea is here derived from the theoretical results. So if we find the best match for each element of the pattern, the best solution, the best cost, the lowest cost will be in the neighborhood for sure. Doing this for all possible matches, we need just to compute the cost using our cost model based on the partial cost, and then we apply, we define an early abandoning optimization that allows us to abandon the computation if we find during the computation a partial cost that is greater than the best cost so far we found. So, as I told you, the overall complexity of the proposed algorithm is logarithmic in the cardinality of the target sequence. This is true under the condition that the cardinality of the pattern is lower, much lower than the cardinality of the target sequence. But this is the case for us, because usually the cardinality of the pattern is less than 10, while the cardinality of the sequence, genomic sequence, ranges between 100,000 and 1 million of regions. So let me provide you a really quickly example. We have a pattern, we have our track. Let's suppose as matching 8 with 15, with root distance 7. Here I highlight a possible matching with the cost 8, but however, as we can see, there is a conflict. So 20 and 22 match with the 27. So we have to enlarge in our matrix of dynamic programming the windows of 1 and find iteratively the good matching that has cost 14 and that makes the matching with 45, 35, 27, and 15. So the good point here to highlight is that we were able to compute the total cost, only computed 6 out of 40 matrix cells. So I just presented you, for simplicity of explanation, the base solution of our algorithm, of our method. During the interaction with biologists, then we come up with several different querying patterns that were so important from a biological point of view, and so we extended our model and our algorithm in order to cover different types of query. In particular, multi-track pattern. So now the pattern is defined on multi-tracks. So we apply the same approach and we repeat it for each pattern track. Negative matching. So pattern track with region that should not appear in the solution. And we do this by removing negative track from the source space before the searching started. We were able to support partial matching, so sometimes matching an element may lead to a poor solution. So in this case, partial match are regions that can or cannot be in the matching. And of course, we had a cost for this partial matching. Further, we also extended the model and the algorithm in order to cover region similarity. So regions are not just point, but can be considered in the side, that can be a type of feature. And in this case, we just extended our formula of the cost model with a dissimilarity between feature of the query pattern component and the element of the track. And finally, instead of provide to the biologists the full result of the total matchings found within the whole genome sequence, we proposed a top K operator that is based on the notion of diversity. So we are able to provide to the biologists only the most representative, the K most representative matching, from which the biologists can then go deeply, let's say using a genome browser, and explore the neighborhood. So from the point of view of experimental validation, we wanted to answer to basically two questions. First of all, is our approach sound from a biological point of view? So we got a real pattern from Polytechnic of Milan, and we evaluated the top K result. Actually, for a limit of time, today I'm focused only in the first pattern, finding and answer regions. And the second question is our algorithm efficient? So for this, to prove this, we used the synthetic data that we simulated the region distances between consecutive region in the pattern and in the genomic track following a Poisson distribution. So let's start with the validation from a biological point of view. Our answer was that represent a multi-track interdistance matching is that we call complex pattern, is composed by two single region positive track, two single region negative track, including a TSS because we want at least a 20K base of distance between each region, and 4 single region as a partial tracks. So the target data came from the ENCODE data repository. We perform the top 100 result that we provide to biologists of Polytechnic Milano that inspected, manually inspected through the genome browser, and they found 100% of precision, so they found all matches of some interesting. To evaluate in a more objective way our result, so the quality of our result, we also decided to compare the whole matchings provided by our algorithm compared with a similar experiment from ENCODE HMM. So here the idea was to find an overlap and to measure this overlap, and what we found was a precision that ranging between 83 and 19. That is a pretty good promising result as a first preliminary real validation on real data from our point of view. Let's keep to the efficiency. For the efficiencies, as I told, we simulated the data. In particular, I'm showing you the time, running time varying with the cardinality of the genomic track that we call with N, and with the cardinality of the pattern side that we call HAM. So we splitted the power of our algorithm in several pieces just to be able to measure the power of each piece of optimization of our algorithm. And in particular, what we found is that if we don't use the Windows-based technique optimization, the implementation is feasible only for limited problem size, up to 1,000. Instead, our algorithm improve with an order of 3 orders of magnitude. And the nice thing is that the early abandoning optimization take the best for a really huge amount of data. And so in particular, when N is 1 million of target region, we save about 70% of the computation time. On the other side, we have just a confirmation from what we found from a theoretical point of view. So the complexity grows linearly with the number of the pattern, the element of the pattern. Finally, as I said, we implemented our algorithm within an integrated genome browser plugin that we name SimSearch, and which allows an intuitive user interaction in both visual selection of interesting pattern on loaded ISCB tracks and visualization of occurrences of similar pattern identifying the whole genome. The desktop application is available on the website, and I provide just a few sketches just to focus on the functionality it provides. Here I'm just running a pattern that is a predefined pattern, so we define the pattern from files, and we finalize just saying that this has to be positive track, negative track. We want even to add a TSS track. We run the search. What we SimSearch return is the amount of the total matching by adding a new line. Each track represents a matching, so an answer. We can zoom in one matching to find the similar match, in this case in human cells. So we found positive, and negative as we wanted in the pattern. So in conclusion, we define a method for finding pattern in the genomic sequences, and we prove the validity, the accuracy of the method through real data provided by Polytechnic of Milano. We define a dynamic programming optimized algorithm for the efficient solution of the problem that is logarithmic in time complexity. The algorithm has been implemented as a plugin of ISCB that is applicable to any genomic transcription region data, and it has great support for understanding biomolecular mechanisms, let's say response to treatments, pathology onset, so on and so forth. And I would like to thank the GenData 2020 PRIM project. I would like to thank you for your attention. I would like to thank you, the organizer, for the special session. A special thank to Stefano Ceri for inviting me. And I would like to invite all the audience to download our desktop application, prove on provided data or your own data, and provide us feedback. Thank you so much.